Morning, everyone. Welcome to the Arthritis Program web seminar series. Today is our lecture 6, and our distinguished speaker today is Dr. Frank Berry from Ireland and Galway. Dr. Frank Berry is one of the leading experts in cell therapy and mechanisms of joint degeneration, as well as in osteoarthritis treatment. Dr. Frank Berry is a professor of cell therapy at Regenerative Medicine at National Institute of Ireland in Galway, and also was a visiting scientist within the arthritis program in Toronto and the member of the academic advisory board within the division of orthopedics within the arthritis program at Toronto. Dr. Berry has contributed to the fields of tissue engineering and regenerative medicine by developing innovative and successful cellular therapies for tissue repair, joint injury, and arthritic disease. He's a coordinator and leading one of the largest trials, which is a clinical trial for stem cell therapy for the treatment of osteoarthritis. It's called ADIPO-8 clinical trial. Dr. Berry is the recipient of Marshall Urist Award for Excellence in Tissue Regeneration Research from Orthopedic Research Society. So good morning, Frank, and welcome. Thank you for taking time to speak today. But before you start your lecture, just a reminder to the audience, please post your questions on the question answer tab on Zoom, and myself will read these questions out to Dr. Frank Berry. Over to you, Frank.
Good morning, Mo, and good morning, Rob, and thank you very much for giving me the opportunity to participate in this seminar series. It's a great privilege to be involved. And you're right, of course, I was a visiting scientist at Toronto Western Hospital in the recent past, and that was a great experience. And I'm very happy to maintain all the collaborations that we started during that time. So I've been working in this field for about the last 25 years. The idea of taking cell therapy, that is a medicine involving living cells, and delivering these living cells to patients as a new advanced therapeutic. And I wanted to tell you a little bit about the sort of strategic approaches that we've taken and how we've been able to use these cells and these models to understand a little bit about musculoskeletal diseases, and of course to develop novel repair strategies. So the topics that I wanted to go through during this presentation are listed here. First of all, how we use stem cells or progenitor cells to give us insights into development and differentiation, specifically in the context of the musculoskeletal system. And because these cells do undergo differentiation processes, this is a good tool for helping us to understand the control of differentiation. Secondly, how we can use pluripotent stem cells, to induce pluripotent stem cells to help us in modeling musculoskeletal diseases, specifically those diseases where the specific mechanistic aspects are poorly understood. Then I'll talk a little bit about the work we've been doing on developing cell treatments for osteoarthritis, talking briefly about the clinical rationale for this. Then I'll do a brief description of the status of preclinical and clinical assessments of cellular therapies for osteoarthritis. And finally, I'll talk a little bit about the nature of the medicinal product that we work with and how critical it is to control this very, very carefully. First of all, mesenchymal stromal cells are point in time. They've been very well characterized and utilized in a large array of different clinical applications. This slide summarizes some of the key characteristics of those cells, which we use in terms of their determining their phenotype. So we rely on specific surface markers, which are listed here, some being positive and some being negative. We rely on the fact that the cells are have trilineage potential. They can undergo osteogenesis, adipogenesis, and chondrogenesis. And then thirdly, we understand that they secrete a large number of very potent bioactive factors, which are important elements in their therapeutic mechanism. In terms of the origin or the source of these cells, they very often are isolated from bone marrow aspirates. They're also commonly isolated nowadays from adipose tissue and from umbilical cord. So there's a variety of sources tissue sources for these cells. We understand very well how to expand them in vitro to generate very large numbers of these cells. And we have phenotypic tests that are used in quality control and as release criteria for clinical use. The first thing I wanted to talk about relates to the fact that these cells are chondroprogenitors. They can differentiate very readily into chondrocyte. Opportunity to understand, to use these cells as an experimental tool to understand how chondrogenesis is regulated, specifically how fate decisions are made by cells in relation to committing to a chondrogenic phenotype. And we've been able to do a number of experiments in this regard over the last few years. And I'm going to tell you just briefly about some of these. So we've been able to develop chondrogenic systems using bone marrow, human bone marrow derived MSCs. We were very careful in how we did these experiments to remove any risk of heterogeneity within these cell populations by preparing clonal lines, essentially by limiting dilution to ensure that we had a homogeneous cell population. And so when we looked at gene expression analysis, this was quite important. Then we used TGF-beta 3 in pellet cultures to induce chondrogenesis in the cells. And the key part of the experiment was to sample at very early time points, beginning 15 minutes after the induction of differentiation, and then going right up to 21 days. And the strategy, overall strategy then was to build an event map based on gene ontology, ingenuity pathways analysis, to try and understand the nature of the events, not so much in overt chondrogenesis, but in the early decision-making process that leads to commitment. So we isolated bone marrow. We took bone marrow biopsies from healthy donors. We isolated the MSCs. We plated these in monolayer culture to the end of P0. And then we generated clonal cell lines from these cells by essentially diluting one cell per well in 96 well plates and looking for those which formed colonies. So the colony forming units generated from these were used to make the clonal cell lines. We characterized them very carefully in terms of the sort of phenotypic properties that I mentioned a few minutes ago, and we ensured that all of the clonal lines that we looked at had the trilineage potential that is the hallmark of these cells. So one particular cell line was very, very successful in terms of trilineage differentiation and was used in this experiment. And then the strategy was really quite straightforward. We take the cells from monolayer, we pellet them at about 200,000 cells per pellet. They're exposed to a medium containing TGF-beta 3, and then they're allowed to differentiate for 3 weeks. At the end of the 3-week period, they're all the very mature chondrocytic phenotype with abundant extracellular matrix proteins. But then we initiate the collection of samples 15 minutes. The cells are exposed to TGF-beta 3, as I mentioned, and then we continue to take these samples at very early time points up to 16 hours. So multiple sampling up to 16 hours, and then allowing the cells to complete the differentiation process up to 21 days to assure that they've reached that mature chondrocytic phenotype. And then the experiment is simple. We do a full RNA-seq genome-wide analysis of the cells harvested at each of these time points. Then we're going to do the clustering to try and understand the time-dependent changes in messenger RNA expression patterns. So we look, for example, where there's rapid downregulation of specific genes, where there's rapid upregulation of clusters of genes at 15 minutes or at 30 minutes and so on. So the time-dependent nature of the clustering is what we look at in the first instance to try and bracket the events that are important at these specific times. And based on all of this, then we can develop an event map based on the gene ontology, the clustering that we see at specific time points, and then using ingenuity and other tools to with the expression of these clusters of genes. So This gives a very, very high level overview of what we see happening in this differentiation process. So within 15 minutes, we see changes in expression of transcription factors, zinc finger transcription factors, genes associated with tissue morphogenesis, genes associated with remodeling of the cytoskeleton, protein ubiquitination, and so on. Within 30 minutes, we start to see changes in expression of genes associated with cytokines, then genes associated with proliferation, and then within 2 hours, those differentiation-related genes that indicate lineage commitment. So it seems that what's happening in the first instance is that the cells are reorganizing themselves, and then within 2 hours, they're becoming committed to the lineage, and then within 4 hours, we start to see molecules that are specifically associated with cartilage formation. And then during the rest of the differentiation period, we see the upregulation of all of the cartilage-specific genes that we know very well, and then overt and abundant expression of all of those proteins into the extracellular matrix. So this gives us the kind of experimental approach, which is really quite straightforward because we're using adult cells, human cells taken from bone marrow. We're able to control the differentiation process very nicely, and we're able to understand the individual steps along that process. Of course, there are many genes that come up that are very, very interesting targets, both for drug development and for understanding the regulation of differentiation, and this is keeping us busy currently. So that's an example of the kind of experimental strategy that many other people, of course, have done similar things, but it gives us the opportunity to uncover some of the interesting aspects of control of differentiation. The second thing I wanted to talk about relates to modeling human diseases, using induced pluripotent stem cells, and what we can do in the context of musculoskeletal diseases to help exploit the extraordinary potential of iPSCs in order to uncover aspects of mechanism or other things. So the idea is very well understood at this point in time. We can take somatic cells, many different cell types from a patient. We can reprogram these cells so that they become pluripotent, induced pluripotent stem cells. And then we can differentiate those pluripotent cells into multiple different cell lineages, and in the process gain insight into mechanisms of the disease or what particular impact the disease phenotype has on specific cell types. We can also use this as a process, as an opportunity for drug discovery, for toxicology assessments, and so on. So, I mean, there are so many thousands of examples of the extraordinary insight that the use of iPSCs gives to different diseases across the whole array of disease types, that it really is an amazing technology. In the context of musculoskeletal diseases, then we can do the following. We can take the somatic cells, reprogram them, and then focus specifically on those lineages which are important to us, such as formation of osteoblasts or osteoclasts, direct differentiation of iPSCs to chondrocytes, or differentiation of iPSCs to iMSCs, and then derivation of chondrocytes from these. And as I mentioned a moment ago, we can use all of these systems to understand the pathology of the disease, or try to unlock aspects of the mechanism. Then as a drug screening or discovery platform, and then for carrying out toxicological assessments. The other value of all of this is that the induced pluripotent cells are essentially indefinitely capable of indefinite proliferation. So we have essentially an endless supply of cells to look at these differentiation pathways and to help in all of these efforts. So I wanted to talk about one particular series of experiments that looking at a type of osteochondritis dissecans. And I think this illustrates very well how we PS technology and help it to shed some light on diseases of the joint or arthritic conditions where we have a need for that additional understanding. So osteochondritis dissecans is well characterized. It's a cartilage disease where there's essentially separation of cartilage from subchondral bone. There's a familial version of this disease called FOCD. It's an autosomal dominant connective tissue disorder where the clinical features include the cartilage defects that I just mentioned, but in addition to that, short stature, early onset osteoarthritis, and in all patients, multiple joints are involved. The particular FOCD that we've been working on, it's sometimes called FOCD NS, referring to northern Sweden, because this is where the condition was first described. It relates to a heterozygous G to A transition in exon 17 of the aggrecan gene, which is a change, a val-to-met change in the C-type lectin domain of aggrecan. And a particular family from northern Sweden was found to have this mutation in all affected members. It's worth recalling for a moment the characteristics of aggrecan, which are important because they do have a bearing on this. Aggrecan, of course, is the most abundant proteoglycan in the extracellular matrix of articular cartilage, and it's essential in terms of the development, sustainability, and biomechanical resilience of articular cartilage. And it's a big molecule, about 2,000 amino acids with some very key and distinctive binding domains. The G1 and G2 domains are at the N terminus of the molecule. The G3 domain is at the C terminus. And in between, there's this long polypeptide chain, which is very richly substituted with chondroitin sulfate and keratan sulfate chains. The N-terminal G1 domain binds to aggrecan, and that's why you see these large supramolecular complexes involving large numbers of aggrecan binding to HA chains. And this is essentially the sort of the polyionic supramolecular structure that helps to give articular cartilage all of the biomechanical features that are important. The C-terminal end of the molecule, the G3 domain, has other protein binding functions, and it binds with very high avidity to a number of other extracellular matrix molecules such as tenascin A, tenascin C, fibrillin, and this apparently helps in the organization of all of this assembly. So when there's a mutation in this G3 domain, suddenly we have an opportunity to try and understand how this controls the assembly of the articular cartilage matrix. So here's the family tree for this particular family, and you can see all the dark symbols are those members of the family that have the condition. And here are two, a mother and her son, from which we were able to obtain both bone marrow aspirates and dermal biopsies. And this gave us the opportunity to isolate primary MSCs and to generate iPSCs from these, giving us then the opportunity to see if we could explore some aspects of the disease and maybe some observations to help understand how this particular mutation in the G3 domain gives rise to this particular set of symptoms. In terms of characterization of the cell lines, we were able to generate 2 iPSC lines from each patient. We also had available to us 2 iPSC lines from healthy donors. So we had a reasonably good experimental setup where we versus controls. We were able to show in the first instance that the from the donors and from the healthy donors and from the patients expressed all of the expected pluripotency genes. And you can see very clearly here in this series of results that the somatic cells do not express these genes, but the do. We were also able to show very clearly that all of these pluripotent cells form teratomas when they were put into a mouse teratoma forming model. And this was very, very clear. Also, their cardiology remained normal despite the reprogramming steps. We were also able to show, and this is very interesting and will have a bearing on what I'm going to say in a moment or two, within we see abundant evidence of formation of cartilage nodules, and you can see these very clearly here, where there's very rich expression of aggrecan, there's rich expression of glycosaminoglycan, and so the chondrogenic propensity of these cells is very, very clear, at least in the teratoma model. The problem comes when we start approaching the chondrogenic propensity of these cells in vitro models. So we were able to characterize then, we were able to generate MSCs from the induced pluripotent stem cells. We also had MSCs from the bone marrow of these patients. And the general observation that we made was that the surface marker expression of the MSCs derived from the iPSCs was essentially as expected. Their capacity to undergo adipogenic and osteogenic difference was essentially as expected, but their capacity to undergo chondrogenesis was modest to say the least. So we then started to look in detail at how we could understand the differences in expression of cartilage-specific genes between those cells taken from the patients, the FOCD patients, and healthy controls. And the broad observation we made, and all this work was done by my colleague Maujia Zhu, Dr. Zhu, who was responsible for all of this experimentation. And this was published in about 3 or so years ago. But the most interesting observation was the following. Look at chondrogenic cultures taken from healthy bone marrow MSCs, or from patient-derived bone marrow MSCs, or from iPSC-derived patient cells. If you look just across here, so there's DAPI staining, there's GRP78 staining, that's an ER marker, endoplasmic reticulum marker, and here's aggrecan staining. And you can see a dramatic difference between the healthy control and the patient, because within the cells derived from the donors, there's abundant extra of aggrecan, very, very clear, essentially uniform distribution of aggrecan throughout the extracellular matrix of the culture. But if you look at the patient samples, either IPS derived or bone marrow derived, the aggrecan distribution is not extracellular, it's either cellular or pericellular. That's telling us that the aggrecan is being synthesized, but essentially it's not leaving the cell. It's not being able to egress the cell to get involved in assembly of the extracellular matrix. And this is one of the key observations, that this disease, because of the mutation in the G3 domain, it's essentially an ER disease associated with ER stress and the failure to process the aggrecan chains. So there's no loss of quantitative expression of aggrecan, but there's a loss of processing of aggrecan through the ER. The consequence of this then is a completely dysregulated synthesis of all of the other components of the extracellular matrix. And some of them are listed here. All of these were done by peptide mass spec, excuse me, quantitative mass spectrometry, where we're comparing the ratio of, that we're showing the ratio of expression of these proteins in patient compared to healthy tissues. And you can see a huge upregulation in some extracellular matrix genes, such as asporin, mimican, downregulation of some others. So essentially there's a dramatic dysregulation of expression of these genes, and there's a failure to assemble the extracellular matrix as a result of either the mutation or the failure to process the aggregate. All of this leads to a particularly weakened cartilage tissue in these patients, therefore the early onset of osteochondritis and the need for joint replacement surgery early in life. So this is an example, and there are other aspects of it that I haven't gone into, but it's an example of how we can use induced pluripotent stem cells to generate patient-specific models and shed some light on what are often complex multi-symptomatic diseases, and we can continue to do this. We have continued to do this using with other disease types and other cartilage and bone diseases. So the features of familial osteochondritis dissecans that we can highlight from these studies are that, first of all, there are normal or near normal levels of aggrecan synthesized in the cartilage of these individuals. Secondly, the key sort of cellular pathology relates to abnormal deposition of within the cartilage ECM and a concentration of the aggrecan in the cellular or pericellular zone, and then a complete loss of control of the synthesis and assembly of other matrix components. So understanding this and understanding the role of the ER processing in all of this helps us to understand the molecular pathology of the condition. I mentioned that chondrogenic differentiation of the process of differentiation of iPSCs into chondrocytes is challenging. And although there are some reports of this in the literature, nonetheless, it remains a complex process. And so what we and others have started to do is look at the pathways associated with development of articular cartilage, either through the paraxial mesoderm or the lateral plate mesoderm, take advantage of what's understood about limb development in the embryo, and then essentially use these signaling processes to stimulate chondrogenic differentiation of iPSCs to chondrocytes. This is what was done by April Kraft and the paper by Oldershaw et al. And so it's a complex series of manipulations of the cells, and I think the efficiency of differentiation is still somewhat uncertain. So we're working very closely on this right now, trying to develop an optimized system of differentiation, but it still remains a challenging process. Okay, so that was a couple of examples of how we're using these cells to understand differentiation and disease mechanisms. What I'd like to do now is talk quickly about the strategies that we've been taking in terms of therapeutic development to understand if we can use these cells to develop a new therapeutic for the treatment of osteoarthritis. The rationale for looking at osteoarthritis is very, very clear. Of all of the musculoskeletal diseases, osteoarthritis is the most common. It represents a very high global disease burden. The direct costs associated with treatment are very, very high and represent a very large proportion of healthcare costs. The indirect costs, the impact on family or patients, their families, and so on in terms of lost income, lost productivity are very, very high. And despite all of this, there is as yet no approved pharmacological intervention, biological treatment, or procedure of any kind that prevents the progressive destruction of the OA joint other than joint replacement surgery. So the need for novel, innovative, probably biological interventions is very, very strong. The rationale in terms of the biological rationale for doing this, sometimes we think of it in the following terms. If you look in all of the tissues that make up the diarthrodial joint, in this case, the knee joint, if you look within the bone marrow, the synovium, synovial fluid, meniscus, cartilage, and so on, within each of these tissues, there's a reservoir of mesenchymal stromal cells. And one hypothesis is that these cells represent the reservoirs of repair cells for homeostasis and tissue repair. And so the idea of delivering exogenously prepared populations of these cells possibly makes some sense. But in any event, this is what we've been doing. We've been looking at models of osteoarthritis. We've been delivering preparations of cells to these OA joints in an effort to understand if there's a therapeutic response. We've done a large number of animal studies over the years, excuse me, and this slide summarizes very quickly the kind of results that we've seen. So this is the meniscectomized, medially meniscectomized goat knee joint Here's a healthy joint. Here's the medial compartment with the meniscus intact. Here's a meniscectomized joint that's untreated. And here's the medial compartment with extensive damage to the articular cartilage, extensive formation of subchondral cyst. Here's the lateral meniscus intact right there. And here's a meniscectomized and cell-treated joint 6 weeks after the cell treatment. So these two are at the same time point. You can see here that the extent of destructive loss of cartilage is much, much less in the cell-treated joint than it is in this untreated control. And there's no evidence of subchondral bone destruction in these cell-treated joints. This was one of the early observations that suggests that cell therapy, delivery of mesenchymal stromal cells, had potentially a structural effect or a disease-modifying effect to either slow down or reverse the degeneration associated with osteoarthritis. And then extensive histological analysis of the tissues in these joints. These are femoral condyles, medial femoral condyles, showing clear evidence of destruction in those meniscectomized and untreated knee joints, and much less evidence of destruction in the meniscectomized and cell-treated joints. And since these studies were done by Mary Murphy and colleagues way back 10 or more years ago, there has been since a huge body of literature involving preclinical assessment of this type of therapy for the treatment of osteoarthritis in every species that's available and in every model of osteoarthritis that's possible. And this table just summarizes, just highlights a few of these key studies. But in fact, if you look at the literature, there are hundreds and hundreds of papers right now looking at the effect of MSC treatment for osteoarthritis. The vast majority of these studies involve intra-articular delivery, just a straightforward intra-articular injection of a suspension of cells. So there's no open surgery, there's no scaffold or device involved. It's just an injection, an intra-articular injection. So in terms of preclinical assessment, there's a very substantial body of literature. In terms of clinical assessment, however, the situation is not nearly as good. And I refer frequently to this paper by Dziwatowski that was published in Osteoarthritis and Cartilage 2 years ago. And this basically involves a systematic review of all of the clinical literature surrounding MSC therapy for osteoarthritis. And their finding is basically that the vast majority of these published studies don't represent level 1 evidence, but or rather represent level 2 evidence. So the database that represents high quality clinical assessment in well-controlled, randomized, fully blinded, placebo-controlled clinical trials is very, very limited. On the other hand, there's very large numbers of small, poorly controlled trials or retrospective studies or different approaches. So there's still, in the clinical arena, very limited high quality evidence and limited long-term follow-up of patients that have received this treatment. There's also, and this is a very serious part of all of this that I'll come back to very quickly at the end, there's an extraordinary lack of consistency in terms of the MSC preparations. The absence of stringent quality control, stringent application of standardization in terms of tests and processes means that very often we're talking about cell preps that have different characteristics. So the poor application of quality control standards to the preparation of the medicinal product that's being tested contributes to the poor quality of the clinical database. And that's something that absolutely needs to be addressed. So in addition to better clinical trials, there needs to be much better attention paid to the quality and characteristics of the product being tested. So the conclusion that we can arrive at this point is that still today, there's no unambiguous or definitive conclusion regarding the effectiveness of these treatments for osteoarthritis. And what we absolutely need are phase 3 studies with rigor attention to design and fully blinded and so on. And that's the direction that we're going in right now. And we're going down this path for quite some time. We're not quite at the end yet, but nonetheless, we are making progress. It's important also to remember that in any clinical trial, for osteoarthritis, it's critical to select outcome measures that distinguish between disease-modifying and merely symptom-modifying effects, because given the complex nature of this technology, the cost of producing these cell therapy products, then a disease-modifying outcome is necessary. And in order to determine in these clinical trials whether there is this disease-modifying effect, there has to be comprehensive structural analysis of repair by imaging of the joints with substantial follow-up of these patients. So to try to address all of these questions, we did become involved here in the ADIPOE clinical studies. And there are two of these trials right now, one completed and one underway. The first was a phase 1 study, which involved 18 patients who received autologous adipose-derived MSC treatment for moderate to severe osteoarthritis of the knee. So the way this works is as follows. The patients come in, they undergo abdominal liposuction, a small amount of tissue is taken, sent to a processing facility where the cells are extracted, purified, and expanded in culture until such time as there's sufficient cells to make a dose. Typically that takes 2 weeks, and then the cells go back for intra-articular injection into the knee joint. The process by which the cells are produced from the fat tissue, it's complex and expensive, and it's a fairly big burden of cost to have a facility that can do this effectively. But in the phase 1 clinical trial, there was a few observations which, although not statistically significant, nonetheless were interesting. There was a rapid reduction in pain scores in most of the patients who received the cell injection. There was also a reduction in, or there was a functional improvement measured using the WOMAC score in the patients that received the cell treatment. Remember, this was uncontrolled, open label, so there's no significance in this. These are merely preliminary observations. There was also no evidence of any adverse events of any kind, so the safety profile of this treatment was looked very good. And so that allowed us to embark then on the ADIPOE 2, which is a phase 2b clinical trial. And although this started a number of years ago, it's not yet completed, but progress is still good. This is a 3-cohort study, 150 patients, 50 receive placebo, 50 receive a low dose, and 50 receive a high dose of cells. The patients come in for a screening visit, and then for 9 subsequent visits. The first visit is screening, the second is liposuction, the third is injection, and then the remaining visits are for follow-up up to 24 months after the treatment. So this study went on quite well up to the end of 2019. There was still a number of patients that needed to be treated. We did manage to put plans in place to complete the trial in 2020, but then with the arrival of COVID, all the clinical trial sites were closed down. The trial was put on hold, and at this point in time, it's still on hold, and we don't yet know exactly when we'll be able to get back on track. So we're still working on this, still not done. It's taken a long time, but we will eventually get to the end point. And so before I finish, I wanted to come back to this issue of product and process control, because it's really very important. So what we're doing in these clinical trials is we're assessing the effectiveness of a medicinal product, which consists of living cells, which are often prepared locally in a cell culture facility under GMP conditions. It turns out that the level of control of this production process is so poor that we really have to doubt whether in different facilities we're even working with the same or different products. The reason for this is pretty straightforward. If you look at all of the process variables, all of the different protocols that people use to prepare these cells, there are a huge number of these variables. Everything from the source of the cells, adipose, marrow, umbilical cord, iPSC, to whether they're expanded or concentrated, the cell expansion media consists of, whether it's fetal calf serum, autologous serum, platelet lysis, and so on. to the vehicle that's used to deliver the cells to the patient, to the dose that's being used. Everything between 2 and 200 million cells represents the dose. Each one of these, I will suggest, is a very potent biological variable. And when you change any one of these variables, you change the phenotype of the cell. And we have plenty of experimental evidence to show that when you do systematically change these conditions, you systematically change the phenotype of the cell. By that I mean you change the cell's capacity to proliferate, you change its surface phenotype, the expression of surface molecules, and you change its differentiation capacity. So each time there is a change like this, there's a change in the phenotype of the cell. So potentially you're looking at a cell preparation with different biological properties compared to the one that's being produced elsewhere. And the testing protocols that we use are insufficient to see these differences and don't respond to these biological variables that I'm talking about. So in order to make the progress that we need to make, not only do we need to do the clinical trials in a very rigorous and carefully designed way, but we also need to pay really strict attention to how we make the products that we're testing, because there's a great deal of work to be done in this regard. I just wanted to mention before finishing, just to indicate the contribution that we've been able to make in the COVID situation. We do have a spin-out company here from our institute. It's called Orbison Therapeutics. It's a cell therapy, it's an MSC cell therapy company. It's located on campus here, and it's a clinical stage company addressing a number of non-musculoskeletal indications, all in early stage 1 clinical trials. But it was in April of this year given approval to initiate a study addressing acute respiratory distress syndrome in COVID-19 patients. And it quickly got approval from the regulatory authorities to use umbilical cord-derived MSCs as a potential treatment for COVID ARDS. That trial started quickly. Quite a few patients were recruited, and it's continuing. So in this regard, we've been able to make our contribution to the search for new treatments for COVID-19. So let me finish there, just to acknowledge the extraordinary input that many of my colleagues have made over the years to all of the work that I've been talking about. Also to acknowledge the funding agencies, and thank you all very much for your attention.
Thank you very much, Frank. That was an excellent overview. So we have a number of questions come in. The first question comes from Jason. With respect to the comprehensive cartilage differentiation experiment, how confident are you that this differentiation is reminiscent of an articular differentiation compared to a growth plate differentiation, as these processes are quite different?
That's a very important question, and it's something that we think about an awful lot. And obviously, if we want to think about articular cartilage repair, we need to have cells that have an articular phenotype and not a hypertrophic phenotype. And that specifically relates to, or amongst other things, the level of type 10 collagen, for example, that we see. We sometimes do see a high expression of type 10 collagen in the primary MSC differentiation protocols that we use. I think in the iPSC-derived models, there's less type 10 collagen, but I think it's still not a question that's fully answered. And I think Jason is right. It is absolutely important.
Thank you. Question from Pratibha. Thank you, Dr. Berry, for an insightful talk. An event map sounded very intriguing as a concept to me. Could you enlighten us on how did you decide with gene selection for a particular go process in the event map? Are they chosen based on how they are differentially expressed?
Yeah, so it's the primary thing is to look at the time-dependent nature of these changes in gene expression. So we look at all of the genes that are upregulated at 15 minutes, at the 15-minute time point. Then we do GO analysis and pathways analysis to understand, are there rational pathway relationships in those clusters of genes that are upregulated at that specific time point. We do the same for genes that are downregulated at the same time point, and also for every other time point. And then that's how we develop this sort of event map. So it's straightforward enough.
Thank you. Another question from Jason that I had too is what the optimal outcome measure tools would be. Jason phrased it as following: do you think that standard of care for patients undergoing MSC treatment for OA would be to include MRI on a pre-scheduled time course to assess the maintenance of therapy, and how might that affect the cost or approval markers at the moment?
Yeah, these are very good points. So yes, there are no good biomarkers, so we don't have an easy test to determine the effectiveness of the treatment. One of the limitations of earlier studies, I think, has been that they only look at the symptomatic aspects. They don't look at the quality of repair of the knee joint. So it's essential that there is an imaging component to the outcome measurements. We're doing both radiology and MRI of the knee joint at the selected time points up to 2 years. That makes it very, very expensive, but for the clinical trials, at least, it's essential. You know, if we can prove the effectiveness of what we're doing, it probably won't be necessary to do this kind of comprehensive imaging routinely.
I had another question, Frank, about it looks like the CD90 marker was the hallmark of the chondrogenic potential of MSCs. Is that correct?
I think, yeah, I'm trying to remember the details of that. I think it's quite important. The problem with these markers, in fact, all of them, is they don't really relate very clearly to the phenotype of the cells, and they don't relate to the potency of the cells. That's why they represent a relatively weak set of release criteria.
Because I noticed on your original table there, it looked like the CD90 chondrogenic MSCs are robust producers of FGF. And I wonder what is at the moment, looking at the landscape, is FGF hold a lot of potential as an anabolic molecule for chondrogenesis?
For chondrogenesis, it's certainly a very important anabolic molecule for MSCs, and it's common nowadays to include FGF2 as a growth factor supplement in MSC expansion. I'll have to check the specific answer to your question, whether FGF2 correlates to chondrogenesis, because I can't recall at this moment.
Thank you.
Congrats.
Oh, here's another question from Somia. Frank, this is an excellent talk. I was struck that in your clonal selection, not all clones give rise to chondrogenic differentiation.
Absolutely.
In your list of variables, how do you rank the heterogeneity within a given MSC population? Should we be selecting for clones with robust differentiation for treatment?
That's one possible. Yeah. So Somia is it's very nice to hear from Somia, and she's absolutely right. There's a great deal of heterogeneity when you do this kind of clonal experiment. You end up with some clonal cell lines which have trilineage differentiation, others have a bilineage, and so on. There's a great deal of heterogeneity here, and you know, in an ideal world, maybe we should be doing this kind of selection. It would add a tremendous amount of complexity to the process, but I think what it's telling us is that we don't yet have the tools that are robust enough to ensure that we're selecting the kind of cells that we need.
Thank you. Question from Armand Keating. Good morning, Armand. Welcome. Wonderful presentation, Frank. Given MSC process variability, is it time to revisit the notion of making a calibration MSC line widely available, for example, from an IPS source, or maybe a DNA ladder as the gold standard that will enable comparisons within clinical outcomes?
Good morning, Armand. It's very nice to hear from you. And yes, I would say that what you're saying, so there's a few ways to think about it. Number one is to develop tightly controlled, standardized expansion protocols so that in essence, every lab everywhere is using identical growth conditions. If we can control the process, then this is a big step forward. It may well be that we could achieve this using an MSC derived from iPSCs. Our understanding so far, based on the experiments that we've done, show that there are considerable phenotypic differences between iMSCs and primary MSCs. We need to resolve that before we can think of using the iMSC as that standard. But I think standardizing the process rather than trying to use standardized markers is probably the way that we should go.
I had a question, Frank, about strategies that might enhance the residence time of an injected MSC population. I assume, like you look both in likely in both in humans and mice on how quickly these injected cells might be cleared and how they might actually be a modification might allow a longer residence time.
Yeah, this is one that has occupied a lot of people's attention. So it's obvious, or it's apparent at least from a number of observations, that the residence time of the cell is quite low, and the efficiency of engraftment is also quite low. So if you track the cells in the knee joint, for instance, they don't last very long, and only a small percentage of them actually persist and engraft for a long period of time. So whatever effect there has appears to be either, number one, it's based on a transient effect, or number two, it's those persisting, that small population of the persisting cells, which are important. We do have ways to think about how to enhance that engraftment using different biomaterials, for example. We did some work in the recent past looking at different polysaccharide, adhesive polysaccharides to enhance the attachment of the cell to articular cartilage. These work very well. We haven't yet been able to test this in a clinical study or in a preclinical study, however, to determine if you do that, if you make the cells stick around longer, do you get a better therapeutic outcome? We don't have that answer yet.
Thank you. A question from Robin Poole. Great talk, Frank. Any insights yet into the reduction of pain by the stem cells in the OA?
That's a very good question, and it really is an important one. And the answer is no, we don't have that understanding yet. It may be associated with a reduction in inflammation, because one of the characteristics of these cells that we do understand is that they have strong immunomodulation properties. We know that they can interact with cells of the host immune system and essentially suppress inflammation. So one thing could be that the cells interact with cells within the delivered cells interact with cells within the synovium, leading to a reduction in inflammation and a reduction in pain. That's speculation at this point because we don't yet have that that understanding.
Congrats on your COVID trial approval. Good luck with that.
Thank you.
Frank, you know, I believe in your clinical trial, you have a lot of lessons learned. And if you're going to go back, what would you change in your trial design? And what would be your optimal design and some of the key points which the audience can learn? Also, the time frame of the trial, I believe you have 24 months currently, where you are looking at patients in a longitudinal follow-up. Is that a good time period for a follow-up?
Thanks, Mo. So those are all very good questions, and we have learned a great deal as we've been going down this road for the last 5 to 7 years. The lessons would be that clinical trials are very, very expensive, and you need to have an assured budget sufficient for that. And if you don't have that, you'll always struggle. So that's why it's so hard for academic consortia to do good big clinical trials, because we just don't bring in the kind of money that's needed. And if you have a commercial sponsor, it probably makes all of this work better, to be honest, because then you have potentially far bigger resources. To do it without the industry partnership is always going to be a challenge. So financial, the budget is number one. It may be that less rather than more centers is better for a phase 2B clinical trial. It's easier to control. So if you have very efficient, very efficient clinical centers that work very well with the patient throughput and all of that, it may be better to have a smaller number of big centers rather than a larger number. Paying more attention to the medicinal product, I talked a lot about that. It's not controlled sufficiently. It's expensive, and there's a lot of variability. I think all of that needs to be looked at very carefully if you're talking about going into a new cell therapy clinical trial. The design of the trial itself and the outcome measures, I think, were quite good, but those other aspects, I think, are important. In Europe also, it's still a complex regulatory landscape. You still have national regulators and you have the European Medicines Agency. So it's not not necessarily an entirely smooth regulatory process in Europe.
Wonderful. Thank you, Frank. It was a great overview, and thank you so much for taking our time today. Again, to the audience, just a reminder, next week we do not have any webinar. Please join us on July 7th for a lecture by Dr. Aileen Davis, on July 14th by Dr. Tohina Nyoshi. So see you all, not next week, but the following week on July 7th. See you all. Thank you, Frank,
Thank you,
for
Frank.
joining us today.
All the best. Bye-bye.
Bye.
Bye.